So, let's try and kind of generically and very generically define a problem in discovering new drugs. Simple terms, it just takes too long to find out whether your drug, your hypothesis and your drug basically work. So think about it this way, the feedback loop is very long. In drug discovery, let's be generalized, the feedback loop from the position, the hypothesis, I want to do this, to finding an answer is as long as 8 years, maybe longer. Generally, those are bad business models. You kind of want to find out quickly, you want to sell things quickly. And then even within that sort of 8 years, things get even more difficult. So you start with, let's say, a hypothesis. Okay, I want to make a drug which cures cancer. Is what's called a small molecule. It can take up to 4 years, 5 years to actually just make the drug, and could cost you $10, $15 million. So, imagine you do that, you wait, now you have to test it in some sort of animal system because you have to have some sort of proof, at least, that your drug is working. And let's say you do, how long does that take? A few years, another, 10, 15, again, very broad range of numbers. But you're already well into the $20, $30 million spends at least, depending, different scenarios. And let's say you get some sort of answer. Now, is that a real answer? Is that give you any comfort that this drug is going to work in a human being? A little bit, but mice aren't humans, so no. Then you get to the next stage. Okay, you've got a hint of, let's say, efficacy, that this drug works. Probably the most important part of drug discovery is what drug regulators are really interested in is this drug safe? That's a very, very important component of it. It takes time, it costs money. So remember, we're still not in human beings. Right? Then you start a phase one. Phase one is kind of what's called a human safety. Phase two, somewhere around the end of phase two for a small molecule, you begin to get a hint of, okay, I've tested it in enough animals and humans and done all that kind of thing, that I'm beginning to see some effect. Okay? There's still a long way to go, phase three registration. So your feedback loop is very long. In simple terms, it takes too long, it costs too much. The whole process just needs compression. We want drugs faster, safer, quicker. We have another parallel system called computers. The mission statement is to take this very long feedback loop and at the very least take component by component and try and compress it in terms of time and cost and get the computer to give you some sort of probabilistic answer. So I've just given one example here where just in the preclinical timeline of a small molecule drug, you're looking at 5 to 7 years. I'll give it a wide range here, $25 to $115 million. And the wide ranges are really because you actually don't know how long it's going to make a drug. It's very unmodular. It's a lot of trial and error with these kind of drugs, right? So sometimes you could do it for 25 million, which is still a lot of money. Sometimes you could do it in 5 years. But a lot of the time I've seen some projects go on for 10, 11 years and never finish. And that's because there's no set menu for doing these things, right? So in business terms, if you just walk away from biotech and drug discovery, that is just about one of the most nightmare business models that you can basically invest in. The upside, the corollary for investors is you could think of these things as a very big lottery ticket. Yes, you can make 1,000 times your money. But really what we want to do in medicines as a society, as a business, is to make it just much more predictable and compressed. We start with the first thing. It's okay, our picks and shovels, the drug component, we can't have it take 4 or 5 years and tens of millions of dollars. What is the best, most validated system out there? And it just so happened for me to be what we were working on at Silence and our RNA drug platform. So maybe one thing to try and understand is how RNA drugs work. So a starting point is COVID vaccines are RNA drugs. So, what happened? A completely novel virus turns up. That virus has got a genetic sequence. We as a society managed in maybe 1 year, which is, you think about the timelines I discussed previously, but in 1 year we managed to compress absolutely everything because we needed to and wanted to. But more than needed to and wanted to, RNA drugs was really the big enabler of it. And that's because we were dealing with a genetic sequence. So what happened was our COVID vaccines are really a process of creating a piece of RNA which looks like the virus. We inject it into humans, and that triggers an immune response, right? Very neat. There was no, if you like, iterations of chipping away and trying to make the drug. We just basically sequenced the virus. We said, look, this is what it looks like. We created the RNA form of it, injected it, and the body did everything else. Very similar to kind of our drug platform, the RNA drug platform. In its most simplistic form, let's say there are 12,000 genes within hepatocytes. Each of those genes has a novel sequence. We can look up the sequence of those genes in a database. So let's say we have a hypothesis which says gene X within hepatocytes is causal in disease Y. Okay, so how do we go about making our drug? As I said, we look up the sequence for gene X, and the way our drug works, and you can think about this along the flavor of computation, we make the opposite code to that gene. So we know what the original gene sequence looks like, we make the opposite code, we, if you like, attach our delivery system, we give an injection, the delivery system takes it round to hepatocytes, it gets internalized, and this is what I mean by sort of the first layer of computation. It has a rule, think about it as a procedure. Opposites attract in genetics, right? So it sees our opposite code, it goes and finds the exact opposite code, which is the gene we're interested in. They match, and then what happens is the cell goes, that matching process means I need to stop that gene X from basically functioning. Very neat. We do nothing. All we have to do is successfully get it into the cell. So the upshot is, if gene X is causal in disease Y, and if we've managed to get our opposite code into there, the cell will stop basically gene X from functioning, which means theoretically disease Y gets better. So you can imagine now that we're in a world of a lot of modular computation. There's all these genes, we know all their sequences, there's a whole load of diseases, and there's basically genes have causes in certain diseases. We know what the opposite codes are. So theoretically, we can stop the function of any gene in hepatocytes. Not theoretically, actually in practice. We've done that many, many times. So our RNA platform has certain pre-built, installed properties, if you like. We know that we can stop the function of any gene in hepatocytes by creating the opposite code. We know that our delivery system always goes to that one place, if you like. We know the cell will do the work for us. We also now know that how long does that gene stop functioning is around 3 months, right? So if you like, a priori, we know how the picks and shovels are going to work, which is a very, very big advantage. If you go back to my previous example of small molecules, they don't know how it's going to work. It's very, what's called empiric, trial and error. Whereas we basically have these tick boxes. And then I guess what it comes down to is ultimately, we can make the drug, the final drug, against gene X in 6 months and for around $500,000. This is as good as it gets, if you like. Okay, so the right-hand side is a fairly typical, what I call performance slide of our drug, right? So you have on the x-axis time. Time starts at 0. You can see the small injection there. So let's say this is actually in non-human primates. So there's a particular gene, you can think about that on the y-axis. At the top, number 1, you'll see that that gene is at maximum function. So remember, we're trying to stop the function of that gene. So generally speaking, it doesn't matter what gene we're talking about, one injection in day 0, and you can see that over a time course period, probably around day 28, you can see that the red line is starting to get almost 90% of that gene has been stopped from functioning. And even go out 84 days, which is 3 months, 50% of it has stopped functioning. So this is what this is telling me is the injection on day 0 is the drug is making its way into the cell, into hepatocytes, and then it takes a bit of time for the cell to basically stop this gene from functioning. 14 days, it really starts to be very powerful, 28 days at its low, and you can see there's a sort of rebound effect, and that's because, if you like, the drug is running out, but still 3 months and half of the gene product, if you like, is stopping functioning. That's a very broad performance that we can see across all genes in hepatocytes, right? So that's, again, what I talk about a priori knowledge of how something will perform. And the benchmark is basically a head-to-head we did with the best competitors out there, again, to show their platform against ours. Remember, what we're trying to do is show that we were as good as anybody else, if not slightly better. You could argue that somewhere, the lower the better, so the red line is a little bit better around day 14 to 28. But really, to show equivalence is really the first pit stop, a very important pit stop. So maybe when you think about the word platform, drug platform, which is platform is a very abused word in biotech, a drug platform should really, in the main, do the same thing every time. So that's a kind of very important tick in the box. We've got 17 patents to kind of protect this. So, what I find that I think I need to repeat is that a couple of things. Not anyone can turn up off the street, or even in biotech, or even in pharmaceutical companies, and be able to say they had access to an RNA platform. The previous slide, that comes from many years of deep expertise, a lot of frustration, et cetera, et cetera. And the second thing that I think I should repeat is that in drug discovery, drug platforms are not to a penny. Again, very different to software and mobile phones. What I've got here is a slide of another drug platform. So maybe there are 3, 4 true drug platforms in drug discovery. This is a monoclonal antibody, and you can see here that this particular drug platform has lasted 40 years, and still to this day is one of the most important drug platforms that are out there. So there's real longevity in drug platforms. The big red arrow you'll see on the screen is where the RNA platform is, where the kind of monoclonal antibody was. 5 approved drugs in RNA. Around 2003 were also 5, and you can see that over 20, 30 years, many, many new drugs were approved. So it goes back to kind of my hypothesis that if you've got a reproducible drug platform which is validated, it's been tested in human beings, and it works generally, they're very important pieces of kit. The next sort of component of the system is, well, what we're interested in is hepatocyte biology. All the data and information to do with biology, genes, diseases within that cell type, really trying to answer the question, okay, so which gene and what disease do we go and try and address with our RNAi platform? I'm thinking about this kind of architecture or drug discovery system in terms of computation. It's very handy that we're in one location within the kind of human body. It allows us to at least, let's say, gather all the information related to that cell. The first thing is, in terms of any business, when any business highly focuses, you would assume that there's a narrowing of your addressable market. Our addressable market is diseases. You asked me a question saying, are we only do liver diseases? And the answer is no. So, here's what I call data structures that we're interested in. The first data structure we're interested in is hepatocytes. So the second thing is, well, roughly speaking, there are 12,000 expressed genes in the liver. We have estimated that there are around 460 genes which are expressed in the liver, which send their products out via the circulation system. And these genes and their protein products end up in very distant organs to the liver. So the liver, by way of its kind of central trafficking system role, you can think about it as a CPU. It processes genes and proteins, sends them out via the circulation. They have effects in distant organs like the eye, even the brain, in particular the heart. What you'll see is then that we are not limited, because of this kind of circulatory system, to liver diseases, but cardiovascular diseases, eye diseases, obesity, NASH, a whole host of rare disorders. So in fact, even narrowing to this one cell type, still has an enormous domain of diseases which can be, let's say, modulated by our drug platform. Our competitors, and the genes they're working on the left-hand side, you can see that the sort of domain of competitors is pretty small. This is a very limited knowledge space, if you like, so a very limited number of people have access to the picks and shovels. On the top of here, you'll see the gene names they're working on. And the crosses say that each company is working on these genes. So what you can see is a lot of congestion, a lot of companies working on the same genes. That's fairly typical of biotechnology and drug discovery. The reason for that is they find the least riskiest things, because obviously the whole industry is so risky. So everyone congregates around known knowledge, right? So an important question for us was, how many genes are implicated in how many diseases? And the number is extremely large. So now the question is, can we apply, let's say, computation to be able to, let's take a total number of 12,000 genes. How many diseases are there? Honestly, probably in the region of 30,000 or so. The answer to that is there are lots, more than there is time and money for us to do, or even the largest pharmaceutical companies. So, application of computation to trying to find these gene disease pairs, as we call them, is one of maybe the main outcomes of what we're trying to do. We were trying to very much avoid that congestion, because in the end you'll come up with the same drugs, then time advantages become very important. So our repository, if you like, for hepatocyte biology information is called HepNet. What is HepNet? It's every scientific paper that you can imagine which is related to hepatocyte biology, all the diseases which we're interested in, all the patterns that we're interested in. In essence, every single piece of information you can find has been stored. It's in the cloud. Maybe what the company was up to a year ago, maybe we even marketed ourselves as a Google of hepatocytes. What did that mean in practice was that even though we had, let's say, all the information in one place, the new information got added, we had a front-end interface, we had some searching functions, we also had some other capabilities to be able to mine differently. Were we really computing the future of medicine? No, because I think we were rate limited by the word search. What did that mean is we have a team of experts, humans. So already, it's great to have humans, you'll always need humans, but to go back to, if you're really trying to compute the future of medicine, you want your computers to do the reading, learning, and understanding, and the humans to be involved less. Let's just put it that way. Kind of what happened around March of this year, it's pretty amazing to think that March this year was such a huge enabling technology, a new, to me, industrial revolution. So here is the concept of generative AIs came around. We've been playing around with them for about a year. Around March this year, there was an exponential, if you like, their functions, their outputs, everything dramatically changed. And that, for our company, for me, finally meant that we really did have a sort of clear line of sight to this kind of computing the future of medicine and a drug discovery system. And if you want, philosophically, we have gone from that sort of search function, trying to develop systems, generative AI models, which can do all that reading, if you like, understanding, reasoning, inferring, self-coding, and automation. Much less reliance on the people, much more reliance on these kind of very large-scale understanding AI systems. I think one day in March I came in and I just said, look, it's a picture, but it's an important picture. We are going to put generative AI systems into absolutely everything that we do. It's the final component to me of the drug discovery system. It's not plug and play. It's not, it's not like Google search. Not everybody has access to it. Not everybody can tailor make their own generative AI models. But as a concept, we put it at the center of absolutely everything we do. Maybe it just is our company, to the point where we want to even add it to our finance functions. Everything, I think, has and will have a component of generative AI and automation to that respect. Question I get asked is, well, why can't everybody else do this? Point at a very important starting point for me is, remember, we started with the very important limited space of RNA picks and shovels, right? So that's a big, big advantage. It's not something that someone can walk in and just, plug and play with. The second component is, people ask me, well, do we use ChatGPT? Well, we do. I use ChatGPT. It's quite good. But really what we mean by integrating generative AI models is much more elaborate than that. What we want to do is fine-tune language models for our own bespoke purposes. What does fine-tune mean is you basically create a language model, and let's say you want that language model to be interested in RNA sequences. You say, all right, I will feed all the information and data on RNA sequences, and I will train that model to be an expert in the syntax and the underlying almost fabric of RNA sequences. It learns the language and everything to do with that particular domain. Then you can apply that to hepatocyte biology. You can create agents for that. You can create an agent for a particular disease. So you can imagine a company where we have lots of different language models interspersed kind of everywhere. So setting up a specialist agent is not just a question of fine-tuning, it's error checking. It's in of itself not something that can be done easily, and obviously we are set up as a kind of hybrid tech bio company with machine learning programmers, data scientists, people you wouldn't necessarily see in a biotech. So you can imagine, let's say, a highly optimized agent, let's say that agent is an expert in hepatocyte biology, you're really asking them to infer information, almost patterns of information that are embedded within data sets and come out with new learnings and potential new hypotheses, right? So you imagine these agents have the ability to self-code, and then you have this kind of agent which, new information, new data that's coming out, it has access to the internet, and that's the way new information comes in. So you can imagine that computing the future of medicine has massively changed from searching to what I'm describing now. One use case that we have is we have the ability, as I said, E-therapeutics can model biology. We can model any biology within hepatocytes within a computer. Well, what does that mean? So many, let's say, biological processes going on within hepatocytes. One is the entire process of clotting. You have a cut. Kind of clotting takes place. And the way we model that is, as you can see, these nodes and this hairball, these are basically genes which are interconnected to each other, and that's, if you like, what clotting, if you like, looks like within a cell. So we have an ability, and this company's been doing that for a long time, to be able to model biology in order to be able to find novel genes which have causes and diseases. And here's an example of a disease, blood disorder, hematology. This is, I think it's the clotting cascade. And what we do is try and find novel targets, within that, and say, look, what's the best place? What's the best gene to go and attack here? Another use case with generative AI is what we call unstructured data. So kind of what is unstructured data? Here I've given an example of patents. Patent is very long. It's a mixture of text, graphics, graphs, tables, chemical matter, something that you can imagine you could search manually, one at a time. Could we, before, have extracted and stored every patent that we're interested in? Yes, we've done that. About now 500,000 RNA patents are residing in the cloud in Amazon. Could we do just some very basic AI? Let's call it basic AI. Did we have a system which could summarize or even categorize those patents? No. Could we easily, for example, get the system to read 2,000 patents and understand the syntax and language and be able to sort of infer things from it? Impossible. No. Could we file new patent applications on new learning? Could the system do it itself? No. It was all manual. Now with the bespoke language models, those possibilities are all possible. All of them. The summary and categorization, very much so. Could we train a model to understand the syntax of RNA patents? All 300 odd pages of text and graphics, each individually, yes. Could we have a world where the system would auto-generate patent filings? Yes, for sure. So, as is sort of colloquially known, large language models are very much about language. But actually, generative AI, which is really the umbrella holding term for large language model, generative AI models can look at any sequence of information. And it just so happens that genes, as we know, have sequences of letters, A's, G's, and T's. And you can see just a sort of, if you like, dummy gene string. This is gene X. And if we just laid out the entire lot of genes in all their strings, if you just look at them as strings, they're two-dimensional, right? But within those, there's all sorts of relationships. So, for example, genes have a beginning function, an end function. They have components of it which have regulatory functions. And there's no system or human being who understands the inner meaning and understanding and syntax of these things. So, this is an area we're very interested in. What are the best places for this opposites attract mechanism within a cell, right? Are there optimal places that we don't know about? Are there regions of genes which basically are causing diseases? And this entire area, again, completely unlocked by the ability of generative AI. And we design and predict our drug entirely in a computer. We're getting close. Going back to this question, can we predict the drug entirely computationally? It's kind of one of our main projects at the moment. So what you'll see is a pre-AI approach, the ETX RNA agent. In the previous slide, I said if we found gene X could be causal in disease Y, we'd go and look up the sequence of gene X, and it would take us 6 months and $500,000 to make the final drug. Fantastic achievement. Can we do this quicker? The answer is this is probably one of the improvements that we've made significant strides in. We are now in a position where we could make a drug in a month. We could make it for 1/10 of the cost. Right? Can we soon get to a point where we make the drug in a computer? I think that's a very tractable and doable thing, and we're well on the way of doing that. And the data here shows, if you just take that kind of far right, the red dot is what we predicted, our model predicted to be the best RNA sequence against a particular gene. When we went and tested it in what's called in a dish, in vitro, and in a mouse, it was actually found to be one of the most active and potent drugs out, that we could basically produce. So this entire area of going from an idea to making the drug has seen almost immediate payback. So how do we make money? We have our, let's call it technology, right? So we have HepNet, the resource depot, if you like, for all related information to hepatocyte biology. Which gene, what disease is the answer, as the question is trying to answer. Gallomic, which is the RNA platform, the know-how, IP. In very simplistic terms, we use those systems to come up with that gene target and disease nomination. And then we say, right, we want to go through that process. We currently have 4 candidates well within that process. But those are 4 candidates which have been found using the systems, going through that kind of, let's say, validation process. Drug design, it says 6 months, then $500,000. We're working to chip that away down to a month. Every component here, animal disease model, toxicology, phase 1, every component we're basically looking at compressing. And this whole drug discovery system is, there will always be clinical trials. You always have to test something in a human being. You probably have to always test something in an animal. But can we get to a place where computer systems, this drug discovery system, can assess the probability of something, let's call it working, a priori, much better than having to wait that 8 years, as I said at the beginning, and finding out the cost of hundreds and hundreds of millions of dollars? I think the answer is yes. So I see a world where this thing gets compressed, the testing in animals and humans becomes smaller, gives a probability, and then you basically can have a much higher probability of success at the beginning of the process. So the next component is, how do we make money out of this? It would be a long time to wait, even with this system, if we had to wait for an approved drug to make money. So there are various different ways of monetizing, doing non-dilutive funding, equity fundings within this entire process. Try to show sort of precedents of what other industry deals have been done. And remember, these are industry deals done without the entire concept of computation or anything like that. These are just general RNA deals. And there have been some pretty big deals done in just the RNA drug platform. So maybe the biggest one and important one for me is there's one for Dicerna and Novo Nordisk. Around 2019, Novo Nordisk approached Dicerna and they said, we want to reserve 30 genes in hepatocytes. In the middle, you can see where there have been licensing transactions, where actual assets. If you show good activity in animals and going forward, there's many transactions there where you can de-risk. Remember, one question is, how many gene disease pairs are there? My answer to that was very many that we could have time and money with. You don't need to necessarily hang on to an asset for eternity. You can hang on to your best ones, or you can de-risk at that component, where you can see some of the transactions that are taking place there. The dotted line is important, because the more human data you get, the more value you can accrue. There are some very large transactions in kind of phase ones in the hundreds of millions of dollars. The idea being that the earlier you do transactions, the less you get, and the later you do transactions, the more you get. But certainly in that 2.5, 3 year period from idea to start of a phase one, there's multiple strands where we can basically do business development deals, and there's very, very large amounts of precedence in that happening. I think on top of that, what really differentiates us is the computation and the drug discovery system. So if you like, within this sort of addressable people that we can go and speak to, these are all very large pharmaceutical companies, you've now got the large tech companies. So the first starting point is that we're a tech bio company. We were set up to merge, if you like, two sectors together. So we have all the staff that you need for a traditional biotech. Chemists, biologists, geneticists, project managers, etc., medics. At the same time, we've got data scientists, software engineers, machine learning programmers, which again are not your kind of typical skill sets that you see in a biotech. And that, I think that's a big advantage for us, because it's not easy to find these people, to, if you like, even train them in the kind of domain of hepatocytes, RNA, drug modalities, etc., etc. It's the culmination really of kind of a lot of different domain experts, all really coming together and with a kind of mission to really change the dynamics of the drug discovery industry. So we have the kind of push and pull of biotech people. At the same time, we have tech people who that really is kind of essential to implement this system. I think we got some, very kind of supportive shareholders who really bought into this kind of, that this is a kind of very carefully laid together drug discovery system which could and will completely change the way this entire industry processes. They're very long-term investors. They understand that they're trying to do something, this is a very big idea, if you like, and will require support and funding going forward.